And to another instance of Biology 121 online. This time we're talking about sexual reproduction, meiosis, recombination, and gametes. Meiosis is a process that is of cellular reproduction that is similar to, but a little bit different from mitosis. So let's just review mitosis, which is the process by which a single cell splits into two identical, what are called daughter cells. There's a four-phase sequence. In this particular set of drawings, prophase begins after DNA replication is already completed. So what we see in anaphase, these single chromosomes. In prophase and metaphase, those chromosomes are doubled as sister chromatids that look kind of like X's when you look at them under a very close microscope. So during prophase, these sister chromatids are assembled at the center of the cell and the nuclear membrane drops. Metaphase, they are lined up down the middle of the cell. During anaphase, they are split from each other and separated. And during telophase, two cells form. Now let's talk about how that's a little bit different in meiosis. The reproduction of DNA happens just like in mitosis. So we have this growth 1 phase just after these cells are originally formed, an S phase during which the cell is synthesizing its DNA, and then a G2 phase where the synthesized DNA in sistered chromatids that look again kind of like X's are here ready to be separated. Now a little bit differently, in meiosis 2, instead of the four sistered chromatids lining up in a single file line down the center of the cell, they end up stuck together with their homologous pairs, forming a group on one side here and another group on the other side to the right. So whereas mitosis, we see all of the chromosomes lined up in a line down the middle, with one side of their sister chromatids facing, in this case, up, and the other side facing, let's call it down, so that they can be separated into an upside and a downside of identical cells. In meiosis, that alignment ends up getting messed up as these sistered chromatids stick together with each other. And what that results in is when the two cells are split, instead of having sister chromatids separated apart so that of these four paired sister chromatids, of these four doubled DNA, we end up with one doubled set down here and another doubled set in a different cell. Again, compare that to mitosis, where if we have these four doubled chromosomes, four doubled chromosomes lined up down the middle, and now we have four single chromosomes going up and four single chromosomes going down. In meiosis, the misalignment during metaphase means that we end up with, instead of four single chromosomes here and four single chromosomes in the other cell, we have two doubled chromosomes in each cell. My guess is that meiosis first came into existence when a cell tried to undergo mitosis and messed it up. And what happened was that cell then said, well, this is no good, let me try it again. And so it goes through an entirely, a whole other set of prophase, metaphase, anaphase, and telophase to split these sistered chromatids. And in the second try, meiosis 2, it successfully splits these sistered chromatids, but now instead of having four normal chromosomes in each cell, it splits into two normal chromosomes in each of four cells. So there's the big difference. Meiosis ends up with these halfway cells. They're called haploids, because hap sounds kind of like hap. Hap sounds exactly like hap. Hap is a word related to the word half. So we get four half cells instead of two full ones. During that same process, there's the reason why these chromosomes ended up stuck together is that they actually kind of wind around each other and overlap. And so this creates this crossing over effect during prophase creates, during the first meiotic division, a mix of DNA from the different parents. So if blue here is representing a female parent and red is representing a male parent, then over in this chromosome, we have DNA from both of the two parents and matching DNA, complementary DNA from both of the two parents over on this other chromosome. And it gets a lot more complicated than that, too. Here we have two sister chromatids. Let's say the one on the left in red and orange is going to be the maternal contribution, the mother's chromosome, and the one on the right is going to be the paternal chromosome. They have X, Y, and Z marked just as locations within those chromosomes. As that crossing over occurs, it's going to be a crossover in very complicated ways. So you see a crossing over at X, another crossing over at Y, and a third crossover at Z. So when these end up, the way that these crossovers end up getting resolved is that some enzymes recognize that these two chromosomes are stuck together, and so they just go in and kind of cut through all of the mess and stick the pieces back together. And when they stick the pieces back together, they end up having these weird mixtures of one parent's original DNA with the other parent's DNA on the same chromosome. And let's look at that sticking together effect a little more in detail. So here we have on the top the sistered chromatids of one parent in darker purple and the other parent in lighter purple. So when we end up with this crossover effect, what happens is, so here's a site of a crossover on these sistered chromatids, right? And remember there's two DNA strands for each. And so when we end up with this crossover, an enzyme nicks or cuts open one strand of the DNA, and then recombination allows us to reconnect that DNA, and then a ligase sticks together the loose ends so that where we had this disconnect, we now are connecting it back up. So the disconnected piece on the right from the second sequence of DNA is being reconnected to the left of the third sequence of DNA, which is from the other parent. And then as we unwind and untangle these, we end up with this recombinant DNA, meaning that it has been recombined from both of the two parents. So if we look at our 4-phase sequence of meiosis again, we start with DNA replication, and then as that recombination is resolved, the crossing over is resolved through recombination, we end up with these 4 doubled chromosomes that each contain both maternal and paternal DNA. And when we think about this, I want to be really careful. So if this is, let's say, my normal DNA, then the blue DNA here represents DNA from my mother, and the yellow DNA represents DNA from my father. As I go through meiosis, the resulting 4 cells contain mixes of my mother and my father's DNA on each chromosome. Now, these 4 cells are going to be, these haploid cells are also what's known as gametes. Gametes are, in mammals, the sperm cells and egg cells that fertilize each other to form a zygote. So from the perspective of my child, they're going to inherit one of these 4 gametes from me and another of these 4 gametes from my partner. So when we talk about the blue and the orange here, from that zygote's perspective, that's grandmother and grandfather DNA that's been intermixed in me. Last point I want to make, if we look at this, this is called a karyotype. It says so down at the bottom, of the 46 chromosomes. So these are human chromosomes visualized under a very powerful microscope. And so we have homologous pairs of chromosomes, and they're numbered from longest to shortest. So chromosome 1 is the longest chromosome, chromosome 22 is the shortest chromosome. And we only get up to 23, we get to 22, and then we have our X and Y chromosomes. Now, X and Y chromosomes are also known as sex chromosomes because they have a very powerful developmental role in determining if the zygote is likely, or fetus is likely to develop as a male or as a female. I want to talk a little bit more about that in the next video as well. We're going to talk about a phenomenon known as intersex, in which there's lots and lots of ways for mammals, including humans, as well as other species of non-mammalian animals, to be neither male nor female at the genetic level, or for their phenotype to not match their genotype. So, just want to give a little forecast and shout out to our non-binaries. We're going to be talking about non-binaryism or intersexism in the next video. It's going to be the focus of the next video. So when we talk about a person with 2 X chromosomes, that person is likely to develop, not guaranteed, but most likely to develop as a female with all of the characteristic features that we think of as female human traits. So that's what we would call a karyotype of XX. And there's 46 because this person has 46 chromosomes. Some people have an extra chromosome on one or another of these. In most cases, that's a phenomenon known as polyploidy. And in most cases, that results in developmental problems that typically result in a failed pregnancy. But in a few cases, there's a couple of well-known instances where a person with a 3rd chromosome, or a 47 chromosome total, develops as a person who can be born. The most common of those is known as Down's syndrome. Now let's take a look at what we think of as the human male karyotype. So we have our 1, 2, 3, 4, all right, all these doubled homologous chromosomes, one of which was inherited from a mother and the other from a father, with a mix of grandparents' DNA in each chromosome. And when we get down to this X chromosome, it doesn't have an exact homologue. It has a tiny little Y chromosome to go with it. So what a Y chromosome is actually an X chromosome, a big chunk of which got lost, cut off. And so a lot of the DNA that would help this individual develop properly is missing. And so instead of developing as a normal female, a human with an XY chromosome develops as a male instead, because males don't have the right DNA to develop as a normal female. I'm going to leave it there for now. Look forward to our talk tomorrow.